What are you doing? We have an exam in 2 hours for immunology.
What do you mean, what am I doing?
You're playing Pacman.
Yeah, playing Pacman is studying for immunology.
How?
Well, if you guys knew anything about macrophages, you would know how similar Pacman is to macrophages.
What?
Here, let me show you. First of all, before I tell you about macrophages and Pacman, let me tell you about the body's immune system. Immunity is an internal defense system. It gives the ability to resist or eliminate potentially harmful foreign materials or abnormal cells inside or attached parasitically to the surface of an animal. The function of the immune system is to block, recognize, and either destroy or neutralize materials that are foreign to the normal self, otherwise known as invading pathogens. It also removes worn-out cells, such as aged red blood cells, and tissue debris, which is necessary before repair can occur. The goal is to identify and destroy abnormal or mutant cells that have originated in the body. Pathogens are primary disease-causing biological invaders against which the immune system defends. The immune response can be divided into two specific components: innate or cellular immunity and acquired or adaptive immunity. Acquired immunity relies on learned responses selectively targeted against a particular foreign material following the first exposure. It retains memories of the attacking pathogens in the environment, takes a considerable amount more time, and takes on one opponent at a time. It requires specific antigens, lymphocytes, T cells, B cells, and antibodies. Today, we'll be focusing on innate immunity and how it involves macrophage phagocytosis.
Innate immunity encompasses an animal's immune response that comes into play rapidly upon exposure by any threatening agent, otherwise known as nonspecific. It does not depend on prior exposure to that agent. Innate immunity is an organism's first line of defense by recognizing pathogen-associated molecular patterns. It consists of skin and epithelial cells and the mucous membrane, making up the following 6 mechanisms: barrier tissues and glands. Inflammation, which is sensed and integrated by mast cells, has primary effectors called neutrophils and macrophages, and basophils to trigger the inflammatory process responsible for allergies. The complement system, which is a group of inactive plasma proteins that, when activated, act as antimicrobial peptides and opsonins. Interferon, a family of effector proteins that nonspecifically defend against viral infection. Natural killer cells, which destroy virus-infected host cells and cancer cells. And finally, symbiotic bacteria, which are countless numbers of resident bacteria found in the digestive system, on the skin, and in the female reproductive tract residue. They can inhibit the growth of invaders either by outcompeting them or by secreting defense chemicals. Innate immunity defends against foreign or abnormal material of almost any type, including pathogens, chemical irritants, and tissue injury accompanying mechanical trauma and burns. All animal types, including sponges, have innate immunity mechanisms. Myeloid cells, including macrophages, dendritic cells, neutrophils, make up an important part of the innate immune system. These cells are called phagocytic cells. They engulf and destroy foreign materials. This is a major form of cell-mediated second lines of defense. Neutrophils are abundant in the blood, quickly enter tissues, and phagocytize pathogens in acute inflammation. Dendritic cells are antigen-presenting cells that process antigen material and act as messengers between the innate and an adaptive immune system. Phagocytosis in these cells is important for the elaboration of a specific immune response, rather than for directly destroying the pathogens. Amoebocytes are wandering immune cells which provide cell-mediated defense, patrol the fluid spaces looking out for foreign invaders. Macrophages exhibit a wide range of roles within the immune system. They are responsible for ridding the body of old and worn-out cells, digesting pathogens, initiating the inflammatory response, and triggering the adaptive immune system through antigen presentation. Macrophages are closely related to monocytes in the blood. These longer-lived cells predominate in chronic inflammation. They also release some important inflammatory paracrines and are found in all body tissues.
The biggest challenge the immune system must overcome is discriminating self from non-self. To overcome this challenge, organisms have evolved receptors that can recognize conserved motifs only found on pathogens. These receptors are called pattern recognition receptors, or PRRs. These receptors target pathogen-associated molecular patterns, or PAMPs. Toll-like receptors, or TLRs, represent another major class of PRRs that recognize a wide variety of PAMPs. TLRs are important in triggering the macrophage to engulf and destroy invaders, like the big dots Pacman must eat before he can eat ghosts. Opsonins also aid identification by tagging an invader for easy recognition by phagocytic cells. Once pathogens have been recognized, they can bind to receptors on the cell surface. This binding induces rearrangements in the actin cytoskeleton for endocytosis. Protein kinase C and members of the Rho family of GTPases help coordinate this process. Once internalized, the pathogen is trapped inside a phagosome. Fusion with lysosomes from the Golgi apparatus then create a phaglolysome. Pathogens are then killed by a diverse range of antimicrobial mechanisms. These include bombarding the pathogen with destructive oxygen radicals, synthesizing nitric oxide, and secreting potent proteases and peptides such as lysozyme or defensin. Hydrogen ion transport can also be used to acidify the internal environment. Once degraded, fragments are either released by exocytosis or move on to antigen presentation.
On the other side of the cell, at the endoplasmic reticulum, the major histocompatibility complex class II, or MHC2, molecule is synthesized with alpha and beta domains. Some degraded microbial peptides reach the MHC class II molecule. Once there, they bind to the MHC molecule at the cleft and are transported to the cell surface where they can bind to T cells in a process called antigen presentation. This results in specific T cell activation and is the interface between the innate and adaptive immune systems. The antigen presentation on the surface of infected macrophages, in the context of MHC class II, in lymph node stimulates Th1, or type 1 helper T cells, to proliferate, mainly due to IL-12 secretion from the macrophage. In addition, macrophages are also responsible for secreting cytokines, small proteins that regulate the immune response. Cytokines primarily act as paracrines, which are regulatory molecules that diffuse locally to neighboring cells in order to evoke a response. Sometimes cytokines can diffuse from the site of an infection to cause a fever. Cytokine release is especially important for macrophages.
See, now do you understand why Pacman is so similar to macrophages?
Ha! High score!
I'll take that as a yes.